Hello friends, welcome to CEC Live Lectures. Dear friends, with our ongoing series on cell biology, today we are going to talk on interaction between cell and extracellular matrix. And for the discussion on the topic, we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Zoology, Hindu College, University of Delhi. Dr. Rawat has immense experience, as his immense experience will definitely help us out in understanding today's topic in detail. So let's welcome our guest, Dr. Varunendra Singh Rawat, and let's try to grab maximum knowledge from him. Hello sir, welcome to the
Thank you. Friends, today I'll be talking about various interaction between cells and their extracellular environment, or the cells with other types of cells. As we all know that the cells are present in the vicinity of extracellular matrix in multicellular eukaryotic organism. Whereas in the case of unicellular organism, the cells are directly bathed in the environment, in the external environment. So what holds a cell in its place? What are the interaction between a cell and the extracellular environment in a multicellular eukaryotic organism? I'm going to talk about that today. So my lecture will basically be covering about the extracellular matrix, the various components which make up the extracellular matrix, and different types of interaction between the cells and the extracellular matrix. So the objective would be to know about what exactly constitute the extracellular matrix in any organism, and what type of interaction exists between the cells and its environment, so that a cell is held in its place by the matrix. Now this particular panel shows us the extracellular matrix, which is beyond the plasma membrane of the cell. And as we can see, there are multiple types of molecules present in this environment. Apart from proteins, there are polysaccharides, and there are fibrous proteins, and there are polysaccharides near the plasma membrane. The carbohydrate moieties which project from the proteins and lipids of the plasma membrane make a layer which is termed as glycocalyx. It is in close apposition with the plasma membrane. Whereas beyond that is the layer which is termed as extracellular matrix, and it is composed of various types of protein. The composition of ECM varies. ECM, that is extracellular matrix, varies from tissue type to tissue type, and the types of protein also varies from one tissue to another tissue. But the basic feature of ECM remains common amongst all the tissue type, and that is it provides support to the various types of cells and a site for attachment of these cells. Now the glycocalyx, as I've told already, is a layer which is very closely applied to the outer surface of the plasma membrane. And this particular layer is made up of carbohydrates which are associated with the proteins or the lipids which are present in the plasma membrane. Along with the carbohydrates, there are certain other materials or certain other proteins which make this particular layer, which is also termed as cell coat. And this particular layer is very prominent in certain cell types like epithelial cells which line the digestive tract. Now this particular layer, that is glycocalyx, is very much important for mediating interaction between the cells, different cells in a tissue, and also between the cell and the cell substratum, that is the extracellular matrix. Apart from that, it provides mechanical protection to cells, and it also acts as a hindrance or barrier to particles which are going to enter the cells, and because of the presence of carbohydrate, it can bind certain regulatory molecules. Beyond the glycocalyx or cell coat is the extracellular matrix, which is a very important component of tissues in an organism. It is important not only for providing structural support to the various tissue type or the cell type, but it is also required for viability of different cell type, because it generates signals which helps the cell to survive and to divide. Now, for example, one of the basic type of ECM, one of the most common type of ECM is basement membrane, which is about 50 to 200 nanometer thick layer of extracellular matrix found in the muscle and fat cell. It basically surrounds the muscle cells and fat cells. It is also present in the epithelial tissue, and the epithelial cells rest on this particular basement membrane. And it is also very common in the endothelial lining of the blood vessels. Now, the function of basement membrane is basically to provide the mechanical support for the attachment of the cells, also it generates certain signals which help the cell in surviving. If the basement membrane is dissolved, or if the protein making up the basement membranes are somehow disintegrated, then the signal which go to the cells are disrupted and the cells are no more viable. Apart from providing a mechanical support to the cells, it also helps in cell migration, because it provides the base or the substratum which helps in the cell movement. Basement membrane also separate adjacent tissues within an organ, and it also acts as a barrier to passage of various types of molecules. Now that was the simplest type of ECM, but ECM exists in diverse form in different tissues and in different organisms. However, the basic component which make up the extracellular matrix are similar in various tissues. The percentage of these components varies from tissue type to tissue type, but the basic component remains same. The ECM is basically made up of proteins which are fibrous in nature, because the fibrous protein provide strength and durability to the environment of the cell. And these extracellular proteins, this ECM proteins, they basically arrange themselves into a three-dimensional network which provides mechanical strength and support to the cells which are present in that particular region. The four basic components of extracellular matrix are collagen, proteoglycans, fibronectins, and laminins. Now, starting with the most abundant protein which is found in humans, which is termed as collagen. About 25% of the protein present in the humans is collagen, which tells us about the importance and the prevalence of extracellular matrix in different tissue types. Now, collagen is basically a glycoprotein, which means that it is a protein which has a large number of carbohydrate moieties attached to it. This particular protein is found only in extracellular matrix. It is not present anywhere else, but it is secreted into the extracellular matrix. Basically, it is synthesized by cells known as fibroblasts, but it is also present in cells of smooth muscle cells and epithelial cells. Now, the speciality of this particular protein is that it is having a very high tensile strength, so that it can stretch to great length without breaking. And this particular feature is very important for its prevalence in the extracellular matrix. Now, this particular collagen family has about 20 types of collagen which have been identified till date, and they can exist in a variety of mixture. That means more than one collagen can be present in a given ECM, and this particular assembling gives a specific feature to ECM of different tissue types. Now, if there are more than one collagen existing together, then it is termed as heterotypic association, and it gives various features like strength and durability to the collagen. Now, each collagen molecule is basically made up of 3 polypeptides. These polypeptides could be of same type, or they could be of different types. And in these polypeptide is a region which is basically a rod-like helical structure, and these 3 polypeptides are woven together like a rope is wound. And because of this winding, it gives tensile strength to the collagen fiber. Now, these fibrils which are made up of 3 polypeptides are arranged together one after another in a staggered fashion, and it makes a very large rope-like structure. And this particular rope-like structure has very high tensile strength because of this particular arrangement of polypeptides in staggered fashion. And this increases the mechanical strength of the collagen fiber. Apart from existing in staggered fashion, there are linkages between the collagen fibrils through lysine and hydroxylysine amino acid residue, and this provides basically a great mechanical strength to this fibril known as collagen fibril. Now, the type of collagen fibril which are present in the matrix, they decide the mechanical properties of the matrix. Now, for example, tendon, which is the connective tissue, it has collagen fibrils which are arranged basically in parallel orientation to the long axis of tendons, which provides a high tensile strength to this particular connective tissue. If we talk about cornea, which is the layer which covers the eyeball, And if we talk about the stroma of cornea, it has collagen fibrils which are arranged in layers. So there are multiple layers of collagen fibrils present in the stroma, and these layers, in each layer the collagen fibril are in parallel orientation. But if we talk about adjacent layers, they are in perpendicular orientation. So it is more like arrangement of wood and plywood. This particular arrangement helps in giving the stroma transparency as well as mechanical strength, because transparency is required for the light to pass on to retina, and apart from that it provides mechanical strength. So basically the arrangement of collagen is dependent on the type of tissue and the type of feature which are required for that particular tissue. Also, whenever there is injury to a tissue and scar tissue is formed at the site of injury, it is basically made up of collagen. Now, collagen is a very important ECM molecule, and if any disruption in the amino acid sequence occur in any type of collagen, it might lead to various problems to the organism because of alteration in the cell type and cell adhesion and tissue type. Now, one example I can cite as mutation in type 1 collagen. If there is a change in the nucleotide sequence of the gene of type 1 collagen, then it can lead to a disease known as osteogenesis imperfecta. Now, in this particular disease, the individual has fragile bones and it has got very thin skin and tendons are very weak. So, any individual who has a mutated copy of type 1 collagen will be suffering from this particular disease, and needless to say, it is very harmful for the individual. Whereas, if mutation occurs in type 2 collagen, then it alters the cartilage of the individual and it leads to dwarfism and skeletal deformities in the individual. Though most of the collagen are fibrillar type, there is a certain family known as type 4 collagen which is not fibrillar, that is, it is not fibrous. This particular family of collagen has non-helical segments, and this provides flexibility, whereas the collagen are known for their great tensile strength. Because of the absence of the helical structure in type 4 collagen, it has a greater amount of flexibility, and this particular collagen type also has globular head, and it is prevalent in basement membrane. If there is a mutation in type 4 collagen, it will lead to alteration in the basement membrane of the tissues, and one such deformity or one such disease which is caused by mutation in type 4 collagen is Alport syndrome, where there is disruption in the basement membrane of the glomerulus, and the ions are filtered which are not necessarily or which are not usually filtered in normal individuals. Now, this particular panel shows us about the collagen. As we can see, there are 3 polypeptides which are interwoven together, and there is a helical region, and these moieties which are made up of 3 polypeptides, they are then arranged together in a staggered fashion, leading to formation of one fiber, and this fiber might have a number of collagen molecules arranged end to end and arranged in a staggered fashion, which gives it a very high mechanical strength. Now, coming to another very important extracellular matrix molecule, which is known as proteoglycan. Now, as the name suggests, it is made up of protein and carbohydrate moieties, and it's basically a complex of various polysaccharides and proteins. Now, the polysaccharide component is basically made up of glycosaminoglycans, which is a type of carbohydrate rich in sulfate group. The oligosaccharide moiety is basically a chain of glycosaminoglycans. This chain is basically a repeating unit of dimer of carbohydrates. Like if we have carbohydrate A and carbohydrate B, these AB units are repeated n number of times to make the oligosaccharide unit. A number of such oligosaccharide chains are attached to the core protein, and several such core proteins are then attached to another glycosaminoglycan known as hyaluronuric acid. Usually they are attached, but it's not necessary that they have to be attached. And this particular arrangement of glycosaminoglycan is it's very acidic because of presence of a large amount of sulfate group and carboxylic acid group which are attached to the carbohydrate. Now, because of presence of a large amount of negative charge, these glycosaminoglycans, they attract cations, and this then attract water molecules. And because of this particular arrangement, the proteoglycans provide a gel-like consistency to the extracellular matrix. And because of this gel-like consistency, they resist the compressing force, whereas the collagen molecules were resisting the stretching force or tensile force. Proteoglycan molecules, they resist the compressing force which might be acting on the tissue. The extracellular matrix of bone is also, it is having a large amount of proteoglycan, but it becomes hardened because of deposition of calcium phosphate salt. So these extracellular matrix molecules are very much important in providing strength or tolerance to compression for various types of molecules. This particular panel shows the arrangement of proteoglycan. As we can see, the orange structure are the proteins, and to it are attached a number of oligosaccharide chains which are gray in color, and these protein molecules attached to oligosaccharides are then attached to a large hyaluronan or hyaluronic acid, which is again a glycosaminoglycan which is non-sulfated. Coming to another type of extracellular matrix molecule, which is fibronectin. It is basically made up of two polypeptides. Each fibronectin molecule is made up of two polypeptides, and these polypeptides are basically arranged in modules, about 30 modules, and these modules are then arranged in 5 to 6 large functional domains. These particular ECM molecules have binding sites for other components of extracellular matrix. They might bind to laminin or collagen, and they also have binding site for receptors which are present on the surface of the cell. And these molecules are very much important in holding the extracellular matrix in a stable attachment to the cells. These molecules are very much important during embryonic development. It has been found that the nerve cells which generate in the neural cells, they migrate to various portions of the body through pathways which are lined with fibronectin. If anti-fibronectin antibodies are injected in the embryo, then these nerve cells are not able to move from their neural crest cell. Also, there are certain organs like kidney and lungs which are formed by a process of branching, and there the epithelial layer becomes divided by a series of clefts, and these clefts are formed in the regions which are rich in fibronectin. So fibronectin, apart from having a role in guiding specific cells to specific sites, it also has a role in molding different types of organs. So this particular panel shows how a fibronectin molecule is arranged. As we can see here, there are 6 domains, and there are sites for binding other ECM moieties like collagen molecules, and there are sites for binding other receptors present on the cell surface. Now another molecule, another moiety which is present in the extracellular matrix is laminin. Laminin again is a type of glycoprotein, and it is made up of 3 polypeptide chains which are joined together by disulfide bonds, and they are arranged together in a cross-shaped molecule. About 15 laminins have been identified in eukaryotic organisms, and they play various roles apart from binding to components of ECM. They also play a role in guiding specific cell to specific destination. For example, primordial germ cells which originate in the yolk sac of the embryo, they are guided to their respective position in the gonads of the organism through the tracts which are lined with laminins. And these laminins, they have specific binding sites for receptors on the cell surface. They can also bind to other laminins. They can bind to proteoglycans, or they could also bind to collagen. It has been found that in basement membrane, laminin forms a network along with type 4 collagen network. As I have already told, they form networks in the basement membrane which are separate, but they together provide a strong mechanical strength. They provide mechanical support to the epithelial cells which are present above the basement membrane. Now this particular panel shows the arrangement of laminins. As we can see here, there is one polypeptide which is blue in color, which is the larger polypeptide, and 2 other polypeptides which are green and red in color. They are wound together in helical structure, and together they give a shape of a cross. Now, another component which is necessary for interaction between the cell and the extracellular matrix is integrin, and this particular molecule is not found in the extracellular matrix. Instead, it is found in the plasma membrane. As the name suggests, integrin, it is an integral membrane protein which has a domain which projects out from the plasma membrane into the extracellular matrix. It has a hydrophobic domain which spans the width of the plasma membrane, and it has a cytoplasmic tails which projects toward the cytoplasm. A number of integrins have been identified, and these integrin molecules basically are present as two polypeptides known as alpha integrin and beta integrin. But the functional unit is made by two integrins, one each of alpha integrin and one of beta integrin. Now, 18 different alpha integrin and 8 different beta integrins have been identified, and though they can give rise to a number of combinations, but about 20 different heterodimeric combinations of integrins have been identified at cell surfaces. Now, these integrins, they are basically required for binding of epithelial cells and muscle cells to the laminins in the extracellular matrix. So, these are the proteins which bind the cell to the cell matrix. These integrin molecules are very important in some function, like forming of clots. Now, the integrin molecules present in the platelets are the ones which will bind to the collagen in a damaged blood vessel and initiate the process of clot formation. So, these integrin molecules play a very important role in various cellular processes apart from binding of the epithelial and muscle cells to the basement membrane. Now, this particular panel shows arrangement of integrins. We can see here there are two polypeptides, alpha and beta integrins, and they have binding sites for extracellular matrix proteins like fibronectin and laminin, and they have binding sites for proteins like talin on the cytoplasmic side. Now, it is suggested that this particular integrin molecule, it exists in active and inactive conformation. Inactive conformation occurs when the cytoplasmic tails of the alpha and beta integrin are in close proximity to each other, and at that particular moment this particular integrin molecule is in a bend orientation. But if there is a signal from inside which separates the two tails of the alpha and beta integrin molecules, then this particular molecule stands upright and it can bind to a ligand which is present in the extracellular matrix. This type of signaling is known as inside-out signaling, as we'll be seeing in this particular panel. The leftmost panel, that is the figure, that is A figure, it shows any integrin molecule which is present in an inactive conformation. But when talin protein binds to it, and this protein binds if there is specific signal inside the cell, when it binds to it, then the two tails separate and this particular molecule stands upright and binds to a green color ligand. Binding to this ligand might activate some other proteins, some other kinases which are present towards the towards the cytoplasmic side of the integrin molecule, and various such molecules like focal adhesion kinases get active, and they then initiate a cascade of steps which can lead to specific signaling pathway inside the cell. So, this becomes the outside-inside signaling. So, integrins are basically responsible for inside to outside signaling as well as signaling from outside to inside, and they exist in two conformations which are basically inactive and active conformation. Now, this integrin protein, apart from binding to proteins of extracellular matrix, they also bind to proteins known as Ig family, which are present on various cell types, and this binding of integrin to such proteins can be easily regulated. Usually, integrin bind to protein which have a specific arrangement of amino acid known as arginine, glycine and aspartic acid domain. And this particular domain is found in all the ECM proteins like proteoglycans, fibronectins, laminins and other ECM proteins. Now, as we know that integrins are involved in formation of clot because they bind to the soluble fibrinogen through this particular domain. There can be generated small peptides which have RGD domain, and they can bind to integrin and thus inhibit its binding to fibrinogen protein, and thus it can inhibit clot formation. Integrins are very much required for influencing differentiation, motility, growth and survival in cells. The normal cells, when they grow in culture, they cannot survive because they are not bound by integrin to the plate, base of the plate. But cancer cell have they can survive even in suspension because they do not they have overpassed the binding of integrin to the base of the plate. But the normal cells cannot survive unless they are bound. They are present on the they are bound by integrin to the base of the plate. Now this binding of integrin to the base of the plate is termed as focal adhesion. Now binding of the integrin to ECM is through specific structures known as hemidesmosomes and focal adhesion. Now what exactly are hemidesmosomes? They are basically found on the basal surface of the epithelial cell, from where they are bound to the basement membrane. Now these epithelial cells are bound to the basement membrane by specific structures which are also known as half desmosome. Herein the integrin molecules attached to one of the ECM protein like laminin or fibronectin and make contacts with ECM. Whereas towards the cytoplasmic site, the integrin tails are attached to keratin, which is a type of intermediate filament. So hemidesmosomes are the site where the epithelial cells are binding to the basement membrane. Towards the cytoplasmic sites are present an array of intermediate filaments, whereas towards the ECM side it can bind to any number of specific ECM molecules. Now this particular panel shows us how the epithelial cells are bound to the basal lamina through hemidesmosome, which are a half coin shaped structure, and towards the cytoplasmic side the tails are bound to a large number of keratin molecules, which are intermediate filament. If there is any problem in the hemidesmosomal structure, if antibodies are generated against hemidesmosome, then it will lead to loosening of the epithelial layer from the basement membrane, and it will lead to accumulation of fluid, which will cause blistering in the organism. Also, if there is a mutation in any of the protein which are present in the hemidesmosomal region, again it will be lead to loosening of the epithelial layer from the basement membrane, and again it will lead to blistering of the blistering in the individual. And this is seen in a case known as epidermolysis bullosa. Now, focal adhesions are basically adhesions of integrin to the plate when the cells are cultured in plate, but they are also found in the regions where where the cells are moving on substratum, like these structures are known as philopodia. So basically herein integrins are joining to the ECM, and towards the cytoplasmic sites are present actin filament. In the case of hemidesmosome, we had towards the cytoplasmic site present intermediate filament, but herein are present actin filament, and these also help in fixing the cell to specific place. And it has been seen that in cancer cells such arrangement it doesn't occur, and focal adhesions are not there. So number of cancer cells can migrate from the original site to various places in the body of the organism. That's all. Thank you.
With this note, thank you, sir. Thank you so very much for giving us this productive session. Friends, we will be continuing further and would be discussing more. You are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Dear friends, as you know that today we are talking on interaction between cell and extracellular matrix, and for the discussion on the topic we have with us in our studios Dr. Varunendra Singh Rawat. Friends, Dr. Varunendra Singh Rawat is a prolific professor. Through his medium today we are getting an in-depth knowledge, and we believe that you are going to grab maximum knowledge from him through this session too. So let's welcome our guest Dr. Varunendra Singh Rawat. Hello sir, welcome to the lecture.
So friends, today I'll be talking about a very important aspect of cell biology, which is interaction between various types of cells. As we know that the organisms can be divided into unicellular or multicellular organisms. Unicellular organisms live in isolation, they are in direct contact with their environment. Whereas multicellular organisms are basically made up of multiple types of cells, different types of cells and a number of cells. What makes these specific types of cells to come together to form a tissue and then organ system is one of the mystery of the science. But this mystery has been explored, and we have a certain amount of knowledge which tells us about how the cells recognize each other and how they come into close proximity and how they hold each other to make interaction so that tissues can be formed and organs can be formed and so on. So today I'll be discussing about various types of interaction which exist between cells in an organism. So this particular lecture will be devoted to various types of proteins which are present on the membranes of various cells and interaction which occur between various cells through these proteins. Now these molecules which make the adhesion of various types of cells together are collectively termed as cell adhesion molecules, and they basically are protein molecules which are projecting outside from the cell. There are protein molecules which project from the cell and bind to the proteins present in the extracellular matrix, and these are known as integrin molecules. Apart from interacting with the extracellular matrix, integrins also can make contact with other proteins projecting out from other cells. So they are responsible for both cell to cell matrix interaction as well as cell to cell interaction. Now these particular proteins are very much important for many of the biological processes like migration of various types of cell during embryonic development, for immune system function, that is providing immunity to organism, for wound healing and many other such functions. Also, these particular cell adhesion molecules are involved in intracellular signaling pathway, which are primarily required for cell survival or cell death or secretion of various types of other proteins. Now, the basic requirement of cell adhesion molecule is that because it is present on the plasma membrane and it is binding some other protein present on some other cell, it has to have a domain which projects outside from the plasma membrane. So this particular domain is termed as extracellular domain, and this is the domain which allows one cell adhesion molecule to bind to another cell adhesion molecule on an adjacent cell. Then it has to have a transmembrane domain which is basically spanning the width of the plasma membrane, that is the hydrophobic region, and then it has to have a cytoplasmic domain in order for it to act as an intracellular signaling molecule. It has to have a cytoplasmic domain which is bound to certain other cytoskeletal elements present in the cytoplasm. So for the cell to get fixed to another type of cell, it has to have extracellular domain which binds to other cell, and towards the cytoplasmic side it has to have a tail which binds to other cytoskeletal elements. Now this particular panel shows us the basic structural features of the cell adhesion molecule. There is a cytoplasmic domain present in the cytoplasm, then there is a plasma membrane domain which spans the plasma membrane, and there is extracellular domain which binds to other such cell adhesion molecule. Now the cytoplasmic tails are bound to other cytoskeletal elements through linker protein. They are not bound directly to the linker protein, but they are not bound directly to the cytoskeletal protein. Instead they are bound to linker proteins which are then bound to cytoskeletal elements. And arrangement of different types of cytoskeletal elements give rise to different types of junctions in various types of cells. Now there could be binding between the same types of cell adhesion molecule, which is termed as homophilic binding, or there could be binding of different types of molecules, which is termed as heterophilic binding, or there could be a binding through a linker molecule, extracellular linker molecule. Like in the case of cadherin family, there is binding between similar type of cadherin molecules of similar cell type, and this is termed as homophilic binding. But if there is a binding between immunoglobulin superfamily protein and integrin protein, which are presented on different types of cells, then it will be termed as heterophilic binding. But if there is a binding between two protein molecules through an extracellular matrix molecule, which is a linker molecule, like in the case of laminin, one protein projecting from one cell type might be binding to laminin, and another protein projecting from another cell type would be bound to the same laminin. So there is a linker molecule between two proteins which are projecting from two cell type. So there are various combinations which are possible whenever we talk of proteins projecting from cellular membrane, and whenever we talk about junctions between various types of cells. Now, 4 families of cell adhesion molecules have been categorized or identified, and these are basically integrins and selectins, immunoglobulin superfamilies, and cadherin families. Integrins are basically made up of 2 proteins, alpha integrin and beta integrin, and mostly they are involved in binding of the cell to the extracellular matrix. These alpha and beta integrin, there are about 18 known individuals, or 18 known types of alpha integrins and 8 known types of beta integrins, and about 20 types of integrins have been identified which are involved in such function of binding of cell to cell matrix. But there are certain integrins which are involved in binding of the cell to other types of cells. Then there is a second major family, which is selectin family, and third family is immunoglobulin superfamily, which is basically belonging to the family of proteins in which comes the immunoglobulin. And then the largest type of family is the cadherin family, which is involved in binding of various types of cells. Coming to selectin family, selectin basically is the name derived from lectins. Lectin is the name given to any type of molecule which recognizes a specific arrangement of carbohydrates. That is, it recognizes a specific oligosaccharide arrangement. First type of lectin molecules were found when lymphocytes were isolated from specific lymph organs or lymphoid organs, and when they were injected back into the organism, they homed, or they went back to the same site from where they were isolated. This suggested the existence of specific molecules which recognized such organs, and the first type of lectin, selectin, which was identified was termed as leucam1, that is leukocyte cell adhesion molecule 1, and it was later on named as L-selectin. A number of such molecules or such proteins have been identified which have the capability of recognizing specific carbohydrate arrangement on different molecules getting projected on the other cell. Now these family of proteins are basically integral membrane proteins, and they are glycoproteins, that is they have specific arrangement of carbohydrate on them, and they are capable of recognizing a specific pattern of oligosaccharide which is presented on other proteins projecting from other types of cells. Now there is a specific structural feature of this selectin molecule. It has of course NH2 terminal, and then it has a calcium dependent C-type lectin like binding domain. This is the domain which is able to bind specific molecules of carbohydrate presented on other proteins. Now as we see here that calcium ions are required for functionality of selectins for binding to other type of protein. And this particular family of protein also has another region known as epidermal growth factor like region, and of course there are a number of repeat sequences present in this protein, and it has a short cytoplasmic region which will bind to other cytoskeletal element present in the vicinity. These type of molecules or these type of proteins are found in number of cell types like endothelial cells, platelet cells, and leukocyte cells. The selectins molecule which are found on endothelial cells, they are termed as endothelial selectin. Platelet selectins are found both on platelets as well as endothelial cells. They are known as P-selectin, whereas endothelial selectin are known as E-selectin. And then we have L-selectin which are found on leukocyte. Now these selectin molecules are responsible for making transient interaction, that is interaction which do not last for very long but for a very short duration of time, which is sufficient for cells like leukocytes to make interaction with the walls of the blood vessels at the sites where there is inflammation and clotting. So leukocytes make interaction with the blood vessels at sites where there is inflammation, so that they can move out from the site, that particular site, to the site where there is infection by any bacterial organism or any other organism, and they can carry out their activity of like phagocytosing that particular organism. But this is not the only not the only place where there is interaction between selectin and other types of tissue. These molecules have also been found to make interaction between the embryo of the humans and the uterus. So interaction between between the embryo and uterus is basically made by selectin. There are of course other molecules also, but one of the type of molecules are selectin molecules. So this particular panel shows us how the selectin molecules are arranged. There is a C-type lectin domain that resembles a head, which will bind to other such molecules present in the nearby region. Then we have epidermal growth factor like domain adjacent to the lectin binding domain, and there are a number of repeats which are made up of repeat units of amino acid, and we have a transmembrane domain and a cytosolic domain which is very small in size, and this particular domain might bind other cytoskeletal elements. So this is an example of P-selectin, but other selectin like E-selectin and L-selectin also have similar type of structure. The basic morphology is same. Coming to second type of family which make interaction between various types of cells possible is immunoglobulin like superfamily, also known as IGSF superfamily. Now, as the name suggests, this particular family of proteins belong to immunoglobulin like proteins. They have domains or structures similar to immunoglobulin. Now, these particular type of proteins, they are not dependent on calcium ions for adhesion between them and other types of protein molecules. Now, these particular protein family, they are found in both invertebrates as well as vertebrates. So it tells us that this might be the origin of the immunoglobulin family. That initial function of this particular family might have been joining of various types of cells together, and later on it might have acquired the function of protecting an organism from other types of organism through immune system. Now, this particular family also has number of modules which are again arranged in various types of domains, and these domains are the ones which interact when there is interaction between one cell type and another cell type. Apart from mediating interaction between the lymphocytes with other cell types like macrophages and lymphocytes, they can target other cells also. That is, lymphocytes have these immunoglobulin like superfamilies, and these lymphocytes can make interaction with other lymphocytes or macrophages, but they can also make interaction with other target cells. 25 different immunoglobulin like superfamily proteins which are involved in adhesion of cells to other cells, that is which are involved in forming cell to cell junctions, have been identified, and there is a big list, and the important immunoglobulin like superfamily molecules are intercellular adhesion molecule 2, also known as ICAM2, vascular cell adhesion molecule 1 going by the name of VCAM1, platelet endothelial cell adhesion molecule 1 going by the name of PCAM1. These are some of the important immunoglobulin like superfamily molecules. Now, there are certain molecules belonging to this family, certain proteins like NCAM and L1, which are not necessarily playing part in immune function. So these molecules play a role in growth of the nerve and synapse formation. It has been seen that during the growth of the nerve cell, the axon cone moves towards specific regions, and for that it requires presence of NCAM and L1 protein. it has been found that individuals which are having mutation in their L1 gene, they suffer from hydrocephaly, which is a fatal disease, which basically is like accumulation of water in the head. So it is a fatal disease which occurs because of mutation in L1 type of protein, which is one of the protein of IGSF. And also it has been found that these IGSF family proteins, they can make interaction with integrins present on some other cell types, like integrin alpha 4 beta 1, which is present on leukocyte surface. It basically interacts with VCAM molecule, vascular cell adhesion molecule, which is a type of IGSF, which is presented on the endothelial lining of blood vessels. So here we see that apart from interaction between IGCF molecules of similar type between different cells, IGSF molecules can also make interaction with integrins of different cell types. So this particular panel shows us how there is linking between molecules of immunoglobulin like superfamily. As we can clearly see here, there is overlapping of domain between IGSF of one cell type and another cell type. So this gives a very strong interaction between these cell types and helps the cell to be in place. It keeps the cells in place. And towards the cytoplasmic side, we can clearly see that these IGSF molecules are attached to cytoskeletal elements like actin filaments. Now coming to the most important and the major family of proteins which are involved in cell-to-cell junction formation is cadherins. Now, cadherins are the most prevalent, as I've already told, and a number of individuals have been identified which take part in cell-to-cell interaction. Now, the size of these protein is about 125 kilodalton, and they are transmembrane glycoprotein. That is, they have a domain which projects toward the extracellular matrix. They have a domain which spans the width of the plasma membrane, and they have a domain which projects into the cytoplasm. And they basically mediate interaction between various cell types, but especially in the cells of epithelium and endothelial cells. Now, these cadherin molecules are dependent on calcium ions for interaction between two types of cadherin molecules. That is, calcium ions, if absent from the extracellular matrix, there won't be any interaction between two cadherin molecules of opposing cell type. Now, they are basically required for linking epithelial cells and muscle cells to their neighbor, but they play a very critical role during the development. That is, embryonic development. They are required for sorting of different cell types, like epithelial cell types are sorted, muscle cell type are sorted because of presence of similar type of cadherin in them. And these proteins, they are special because they do not interact with extracellular matrix, and they only interact with their similar type. That is, the interaction between cadherin is homotypic interaction. The structural arrangement of cadherin molecule is such that about five domains project out from the plasma membrane into the extracellular matrix. So if there is an interaction between two cadherins from opposing cell types, then there will be overlapping between the domains which are projecting outside. Now, the interaction could be between only one domain, it could be between two domains or three domains or five domains. Greater the amount of overlapping between the domains, greater will be the strength of attachment. Suppose two cadherins are overlapping in 5 domains, and in another scenario, two cadherins are overlapping in only one domain. Then the region where there is overlapping between 5 domains of the cadherin will be of greater strength. That is, the interaction will be of greater strength as compared to the region where there is only overlapping between one domain of the two adjacent cadherin. Now, I've already told that these interaction between cadherin molecules are homotypic interaction. That is, one type of cadherin will only make interaction with similar type of cadherin which is projecting from some other cell. And the cadherin molecules, when they are present, they are bound to the specific cytoplasmic proteins like catenins, and of which there is a number of types like alpha catenin, beta catenin, and gamma catenin. The cytoplasmic tails of cadherins are bound to proteins known as catenin, which are then linked to cytoskeleton molecules like actin or intermediate filament. This particular panel shows how cadherin molecules are linked together and how they make attachment with each other, making important cell-to-cell junction. Here we can see two cell types, cell 1 and cell 2, and we see cadherin molecules with 5 domains projecting from each cell type. These are similar type of cadherins and they are making interaction at 3 domains. Towards the cytoplasmic site in the lower panel, we can see there are present proteins like beta catenin, alpha catenin, gamma catenin, and one type of unidentified protein is also there. So the cytoplasmic tail of cadherin is making attachment with a complex of proteins which involve catenin, and these proteins are then making interaction with microfilaments, which are basically an assemblages of actin molecule and a part of cytoskeletal element. There are a number of types of cadherins have been identified, and the classical types which have been identified are E cadherin, which are basically epithelial cadherins, P, which belong to placental cadherin and epidermal cadherin, N, which are found in the neural cells, and VE, which are vascular endothelial cell cadherin. But there are also other type of cadherin which differ in the domain arrangement from the classical cadherins, and they are known as non-classical cadherins like desmocollin and desmoglin, which are usually found in the desmosomal regions of epithelium, and protocadherins which are found in synapses. Now these were the type of proteins which are required or which make different types of cell to cell junction. Now what are the type of cell junctions which are present between various cell types in the animal tissue? Now these cell junction could be anchoring junction, like as we see in the case of hemidesmosome, which are junction between the cells and the basement membrane, or there could be desmosome or macula adherens, or there could be occluding junctions which prevent passage of molecules from one side to another side between epithelial cells. That is, no molecule can pass between the space which is present between the two cell types. Then there are junctions which help in communicating, and these are known as channel forming junctions. And there are of course junctions which are involved in passing of signal like synapses. Now coming to different types of junctions, the basic adherent junction, that is junctions which make attachment of one type of cell to another type of cell possible. First one is adherent junction, which is also known as zonula adherens. This is usually found toward the apical side of the cells, and mostly it is found in epithelial cells which line the intestine, and that is present toward the apical surface. It forms a type of belt around the cell, and this belt is attached to a number of other cells which are present in the boundary of that particular cell. This particular type of junction is formed between extracellular domains of cadherin molecules, that is similar type of cadherin molecules projecting from similar type of cells make contact towards the extracellular region, but towards the intracellular region, that is the cytoplasmic region, this particular type of molecule has attached actin filament or microfilament. These type of junctions are very much important for transmitting important signals which help in the survival of cells. This particular panel shows how a zonula adherens or adherent junction is arranged. We can see here overlapping of cadherin molecule with each other, and towards the cytoplasmic sites are present actin filaments through which are attached to the cytoplasmic tail of cadherin through beta or gamma catenin. Another very important adhering junction is desmosome, also goes by the name of macula adherens, which is a circular type of junction, and it produces very strong adhesion between two cells. They are disc shaped in structure, and they are usually found in tissues which are subjected to mechanical stress like skin and intestinal epithelium and heart muscles. Basically, here also we have cadherin molecules projecting from the two types of cells in the binding together in the extracellular region. But here the cadherin molecule which are present, they are a bit different from the classical cadherin, like desmoglein and desmocholine are present here, and they are attached to the intermediate filament. Instead of keratin filament, they are instead of the actin filament, they are attached to keratin intermediate filament towards the cytoplasmic site by through some other linker protein. So basically this structure has outer dense plaque in the cytoplasmic domain, wherein the cadherins are attached to placoglobin and placophilin, which are then attached to desmoplakin, and inner dense plaque where desmoplakin attaches to the intermediate filaments of the cell. If there is antibodies are produced against desmosomal protein, then it might lead to severe blistering of skin, as was found in case of hemidesmosomal antibodies. So this particular panel shows how desmosomes are arranged. There is overlapping of cadherin molecules, and towards the cytoplasmic site are present keratin filament. Then one other important junction is tight junction, which is also known by the name of ZONULA occludens. As the name suggests, it occludes or it prevents passing of molecules or ions between two cells. And how that happens? Because this particular junction seals together the two cell types so that nothing passes between the two cells. And these junctions are basically they encircle the cell like a gasket, so that nothing passes from the apical end towards the basement end. Now these ZONULA occludens are basically made up of several integrin proteins. Initially it was thought that they are made up of protein known as occludin, but later on it came to be known that other type of protein known as claudins are also present here. About 24 types of claudin have been identified, and though earlier it was thought that there is no passage of anything from one side to another side of the tight junction, but recently it has been known that certain types of junctions allow specific ions to pass through. Like for example, tight junction present in the ascending limb of loop of Henle, there are present specific molecules, specific protein known as claudin 16, and these claudin 16 allow Mg2 plus ion to pass through them. Individuals which have mutated claudin 16 gene, they have low amount of magnesium in their blood because magnesium cannot pass through such mutated proteins. So though tight junction do not allow most of the molecules to pass through them, there are tight junction which will allow specific molecules or ions to pass through them. This particular panel shows arrangement of tight junction between two types of cells, making specific boundaries between towards the apical region of the cells. These junctions provide strength and stability, and they are also responsible for maintaining cell polarity and providing barrier in the brain region, blood-brain barrier, and they also prevent loss of water from the epidermis. Then another important type of junction are the communicating junction, also known by the name of gap junction, which allow specific molecules to pass between different cells. In animals, only about 1000 Dalton size molecules can pass through, and wherever gap junction are present, the intercellular space narrows from 25 nanometer to 3 nanometer. And these gap junctions are basically made up of proteins known as connexins. And 6 connexins come together to form one connexon, which is a pipe-like structure. Of the opposing cells, 2 connexons come together to make a continuous pipe so that molecules can pass from one cell to another cell easily. And these connexins can be formed, there are a number of types of connexins, and number of different types of connexin can come together to make one connexon. So these are very much responsible for passing of important molecules like cyclic AMP through different cells. And in plants, there is similar structure known as plasmodesmata, which allows passage of such molecules, but the size is bigger. It can allow about 50 kilodalton molecule to pass through them, but it is made up of desmotubule, which is derived from smooth endoplasmic reticulum. Thank you.
With this note, thank you, sir. Thank you so very much for giving us this productive session. Friends, if you have any feedback or questions, do write to us at info.cec@nic.in. The lecture is going to be uploaded on YouTube soon. So keep watching us, keep writing us. We will be meeting again soon. Until then, take care. Goodbye. Thank you, sir. Thank you once again.